Hi, everybody. I'm Tomaz Berisa. I'm the CTO at Gencove, which is a company trying to make genomics more accessible and interpretable by getting low-cost sequencing into the hands of as many people as possible. Today, I'd like to talk about Gencove's approach to sequencing genomes and the technology and architecture that we implemented to support that approach. When choosing a technology for a genomics project, one is often provided some variation of the choice between sequencing and genotyping. The industry gold standard, whole genome sequencing, reads a whole genome around 30 times over for increased confidence and is great for discovering new genetic variation. On the other hand, genotyping arrays enable researchers to confidently measure very simple mutations that have been chosen in advance, most likely because they're known to have a certain effect. The main rationale behind this choice is a trade-off between comprehensiveness on the x-axis and price on the y-axis. The whole genome sequencing is comprehensive, but quite expensive and relatively low throughput. And as a consequence, users still rely on legacy technologies like genotyping arrays that are limiting for genomic discovery. And this is especially true for population studies where total study cost is a limiting factor. Ideally, we'd have something at the cost of a genotyping array, but sequencing based and relatively high throughput. Our solution to this is low-pass whole genome sequencing. It provides over a 10-fold reduction in cost per sample relative to whole genome sequencing. Its price is lower than or equal to that of a genotyping array, and it provides 10 times more data. It has new variant discovery capabilities and does not exhibit ascertainment biases across different populations. To give you an idea of how low-pass sequencing works at Gencove, I will compare it to whole genome sequencing and genotyping arrays. Low-pass sequencing takes a relatively small random sample of the genome and then fills in the blanks using a method called imputation. Imputation is a statistical method for replacing missing data, and for our use case, it consists of an algorithm and a reference panel of high-quality genomes. By comparing a random sample of the genome to the reference panel, the algorithm is able to identify similarities and fill in the blanks with best guess estimates. Much like your knowledge of the vocabulary and grammar of the English language enables you to fill in the blank of the following sentence. To say, I saw a blue cap on your head. Of course, it's a simple example, and most commonly there's uncertainty in these estimates, like in the following sentence, which is slightly modified towards the end to provide less context, and the answer could be any of cab, can, cab, car, cat, with varying measures of uncertainty in each of these guesses. So properly quantifying these uncertainties and utilizing as much context as possible is at the core of our work at Gencove. Also, I have to mention that these great examples are originally by our friends Yaniv Ehrlich and Asaf Gordon at DNA Land. To provide more real-world data on the for the genomics experts in the audience, I'd quickly like to go through some of the results of our work with GlaxoSmithKline, which was led by Charlie Cox on their end. They were curious about how imputation from low-pass sequencing data compares to imputation from genotyping arrays. 79 European ancestry individuals were sequenced by Gencove to 1x coverage, and then we downsampled that to 0.8, 0.6, and 0.4x, after which we ran our standard analysis pipeline. And the same samples were genotyped by GSK on the Affymetrix PMR array, which has around 800,000 SNPs. They took these two data sets and compared them on their end. The first analysis they came up with is a sanity check to check the concordance of low-pass sequencing data, assuming that the genotyping array is the truth. And the part of interest here is the non-reference genotypes, because that's the part that's hard to guess. The accuracy at 0.4x was around 98.2%, and the accuracy at 1x was around 99.2%. Accuracy is far higher than that at reference genotypes, but that's kind of expected because it's not hard to guess the reference genome. Furthermore, we compared the R-squared of imputed genotypes to truth for genotyping arrays and low-pass sequencing across different allele frequency bins. We can see that the 0.4x sequencing data approximately performs equally to the genotyping array, and adding more sequencing just increases the study power of the experiment. And the advantage of low-pass sequencing is even higher in non-European populations, where genotyping arrays exhibit high ascertainment biases. In addition to imputation, we developed a suite of algorithms and tools to deal with low-pass sequencing data, like ancestry analysis, disease risk profiling, relatedness detection, etc. All of the mentioned analyses are provided as an end-to-end service from sample or DNA all the way to delivered data. And as you can see, our products are competitively priced to genotyping arrays. And for larger customers requiring very high accuracy variant calls on specific genomic regions, we can also include an additional capture step for a list of predefined genes for a minimal price increase. And for users with custom regulatory, geographic, or other requirements, we've developed a sample prep kit so they can process samples in-house, run it in their own lab on premises, and use our analysis software as a service to make use of the resulting low-pass sequencing data. Common users of our technology are pharma companies, biotechs, and research universities like GSK, Calliope, Stanford, and Harvard University. And all of the mentioned functionality is wrapped into an API and provides our clients with programmatic access to their low-pass sequencing project. Today, I'll provide a brief overview of this part, our primary and secondary genomics analysis pipeline. An overview of the workflow from sequencer-delivered data is outlined here, with stars denoting in-house analysis tools and algorithms for processing low-pass data that we had to develop in-house. In theory, it seems straightforward, but when taking into account our standard run on the Illumina NovaSeq yields data for 1,500 samples at a time, and we run these fairly oftenly, you can imagine things can quickly get quite complicated. Luckily, AWS provides us with a reference architecture to point us in the right direction, and for anybody running this workload or these types of workloads, I highly recommend this read. But as one always does, we needed to modify this architecture to fit our needs, and I'll provide a quick overview of modifications in the following slides. The architecture relies on running HPC jobs on AWS Batch and orchestrating them with step functions in Lambda. As we all know, Batch is a service that provides managed batch processing. Step functions provide serverless orchestration, and we all know Lambda enables us to run code without provisioning or managing servers. We start with programmatically describing the workflow, which is naturally done in step functions. Here I show a one-step workflow to keep things simple, but in reality it has many steps. You can think of it as a representation of the analysis workflow outlined earlier in the talk. When a step is started, the details are submitted to an activity task queue. This queue is long polled by another step function, and it submits the required jobs to AWS Batch. Long polling the queue enables us to control the rate that jobs are submitted to Batch, which in turn turns out to be an important parameter and the first key component to running these types of workflows in a highly parallelized fashion. Because overwhelming the Batch API with many parallel submit jobs did not work well in our experience. And therefore, being able to rate limit this has been a big win for us. Once a job is completed on Batch, a CloudWatch event is generated to trigger a Lambda and unblock the execution of the step function for it to continue on to the next step or complete execution. The event-driven aspect to this architecture is the second key component to running these workloads in a highly parallelized fashion, because constantly polling the Batch API with described job requests to get the status of jobs has also not worked well for us, especially because this can be avoided altogether using the outlined approach. In conclusion, I can report that the modified architecture enables us to run our workflows without issues, and the only thing we have to do is submit service limit increases from time to AWS. I'd like to take this opportunity to thank the folks from AWS, Paul, Angel, Alexis, and Elliot for their help, and a big thanks to the Gencove team. A more detailed write-up of the modified architecture together with source code and infrastructure templates is available as a guest post on the AWS blog. And also we're hiring, so please spread the word if you know great engineers excited about genomics. I'd like to thank you for your time and attention, and happy to take any questions now, or please feel free to grab me later on.
Is there a genotyping step with all the samples during the genotyping? It's kind of standard what you do with high
Genotyping? Or joint imputation?
No, so just taking all the reads, lining them up from all samples.
Yeah.
Jointly genotyping across all samples.
Yeah, so what we do that mostly for variant discovery. So when users when our customers will have a large study and they sequence thousands and thousands of samples, we will put all the reads together, and that's how we enable them to discover new variants in the